Welcome, everybody. This is UC Davis, the West Coast Metabolomics Center monthly seminar, supported by the Genome Center focus group on data science in metabolism. Today it's my great pleasure to welcome Professor Gaurav Moghe um, to be our speaker today. Gaurav Moghe has an illustrious career and definitely will talk about things that we haven't heard before. So it will be very new to us in a way. And it's very well aligned with the idea of data science in metabolism because that's what he does. He did his Bachelor of Science in Ramnarayan Rua College in India in microbiology, 2001 to 2004. Then he did his master's at the University of Baroda, also in India, from 2004 to 2006, with a thesis title on gene content evolution in plant genomes, including whole genome duplication and intergenic transcription expressions in brassicaceae. Then he went to the United States and where he got his PhD at Michigan State University in genetics from 2007 to 2013. He stayed there. He must have liked it in Michigan, East Lansing. So he went then to East Lansing, where he did a postdoctoral research from 2013 to 2017 using multi-species transcriptomic and metabolomic analyses, including acyl sugars. So these are interesting compounds that are expressed by certain cells of trichomes in plants. So these are really cool, compounds. And the related to plant defense, and he looked at Solanaceae here. And then finally, with all that preparation, he got his faculty position at Cornell University, College of Agriculture and Life Sciences, where he started full-time in 2017. And he was assistant professor for 8 years, so 2017 to April 2025. And last year, well, he did computational biology on and biochemical genomics there. And last year, he was promoted to the level of associate professor at Cornell University. Congratulations to that promotion, Gaurav. And so we are very much interested in hearing about metabolomics, metabolites, plant genomics, and the function of all of that in the roles of these compounds in plants. So the floor is yours.
Okay. Everything showing up okay?
Yes, we hear you. You can be a little louder, say something again.
Yeah, I think, how about now?
Yeah, that's a little better. Okay.
Okay, the speaker sometimes gives me a problem. Okay, thank you, Oliver, for inviting me to give a talk here, and to Janet for organizing all the logistics of this seminar. So I'm going to talk about plants today and our use of various approaches for understanding how plants respond to stress, and how are their pathways governed in terms of their enzymes making those plant metabolites. So before I start, I wanted to advertise a seminar series that I myself organize along with a few others. It's called Phytochem Talks. This is held every Friday. Our next session is this Friday. And we've been organizing this since September 2021. And we've had a lot of diversity of speakers over the years, not just from plants, but also from other species. And this Friday, we have a talk by somebody by Dr. Sushma Naithani, who works on plant reactome database and how that can be used for gene discovery in phytochemistry. So if you are interested, please register on this Google group and you'll get invites. I would also like to start by acknowledging the people who have done the work that I'm going to talk about, as well as Frank Schroeder and George Gender here at the Boyce Thompson Institute, who graciously allow us to use their Orbitrap instrument for our experiments, and funding through various organizations. I'm going to first give a short two-slide motivation for the work that we do. And my research talk is focused on two different aspects of our work. First is how do we look at plant LCMS data and interpret, how do we improve the interpretability of those of the features that we get. And second is looking at enzyme function. Okay. Now, I don't need to tell it to this audience that metabolites govern all processes in organisms and plants too. There are fundamental aspects to the processes governed by these metabolites, ecological aspects, development, stress adaptation, etc. And they have importance in both fundamental science as well as application oriented. And that's what, after doing a PhD in genomics, that's what got me interested in plant metabolism, where I did my postdoc and spent hours in front of water Skewtoff at Michigan State. So plants produce a huge diversity of metabolites. It's estimated that there are over 1 million metabolites that are present across plants, and that's due to the complexity of the underlying metabolic pathways. We typically use LCMS, right, for identifying these metabolites or for detecting these metabolites. But we all know that metabolomics it does not capture the whole metabolome. This is one of the challenges. First of all, the extraction method that we use biases us against the type of metabolites that are isolated, solvents that are used, the type of mass spectrometry, the column chemistry, all of that, and the LC and MS methods, of course, can determine what sort of metabolites are detected finally in our data. Secondly, among the features that are detected, most of these features have poor annotation. So it's estimated that there are 1 million plant metabolites, but based on several papers that we looked at, very few of them have actual documentation of the structures, and many, less than 10% of the peaks in tomato could be annotated using database searches. So over the last few years, one theme of the research in the lab has been how can we improve the identification of these metabolites using the MSMS fragmentation patterns. One class of compounds that we work with are these compounds called resin glycosides. They are type of acyl sugars. So acyl sugars that I worked on during my postdoc are primarily present in Solanaceae species, tomato, potato, and other species. But these resin glycosides are produced only from sweet potato and related species. They comprise of this oligosaccharide core, to which there are small acyl chains being attached, as well as this long C16 fatty acid with the 11 hydroxylation that kind of sometimes circularizes, sometimes does not. And these compounds offer protection against nematodes to sweet potatoes. They also have medicinal properties, and they have bioactivities against bacteria, fungi, insects, etc. Now, if we know the structure of these compounds, we know that we can assign specific moieties to different fragments that we get. And previously we used to do this one by one by looking at individual features. Then we started, if we know this 271 peak corresponds to the C16 fatty acid with the hydroxylation, there can be C14 hydroxylation, C18 hydroxylation, and then you could have a glucose attached, a pentose, a hexose, pentose, methyl pentose attached to it. That will give different fragmentation patterns. So using this in silico library of all possible fragments for the signature MSMS fragments, we looked at over 30 different species of the Convolvulaceae family. And just to give you a scale of this analysis, since 1992, when this class of compounds have been discovered, over 250 structures were identified in different Convolvulaceae species. But by looking at it computationally through these fragmentation patterns, we can identify 330 resin glycoside-like peaks in just one species. So there's a huge diversity which remains hidden if you're not able to capture those that diversity. And we can also look at identify what sort of sugars are present, what sort of long chain fatty acids are present, and then what sort of acyl chains are also present in each of these species. Previously, we had a set of Python scripts that would go through the MGF files produced by MS-Dial and produce this kind of an output or identify the resin glycoside-like peaks. But now you can just upload those MGF files to our website and identify all the resin glycoside peaks. My students, without computational experience, can do this very rapidly. With that, we can have we get access to evolutionary insights. So this is the molecular network of all the peaks obtained from all of the species, grouped by genus, genera. And we can see here that there is the convolvulus species have a different sort of resin glycosides compared to the Ipomia species. Those are the two biggest genera in the Convolvulaceae family. And looking at the masses of these compounds, we see that the Ipomia species produce more low molecular weight resin glycosides, and the convolvulus species produce more high molecular weight resin glycosides, which we could attribute to additional additional sugars being added to the oligosaccharide core, as well as some different small acyl chains that are added to the structures. Now, we are looking at this in more detail in different sweet potato varieties too. So, Bhaswati is a grad student in the lab. She's working with 40 different sweet potato varieties and trying to identify where these resin glycosides are produced. We see them produced near the periderm, which offers protection against the nematodes as well as the fungi. And then Mohammad Irfan is a postdoc in the research associate in the lab looking at which enzymes are responsible for the biosynthesis. We think we have, so we identify the enzymes using RNA-seq, proteomics data, and then perform enzyme assays. We have to do virus-induced gene silencing to make sure that it's not a promiscuous activity or express them in Nicotiana and ensure that the enzyme is doing the reaction that we expect it to do. So we've identified a couple of enzymes and we're preparing a paper for that currently. Now, if we have the MSMS fragmentation pattern, if it's known, then we can look at these classes in more detail. I mean, that is a constraint, right? We've done the same thing for anthocyanins and flavonoids, where the fragmentation patterns can be generated or analyzed in more detail. This tells us that the orange flesh sweet potato lacks many of the anthocyanins that the purple flesh sweet potatoes produce, but it has several of the flavonoids that are present, but not the anthocyanins. We have done the same thing in acyl sugars in tomatoes, wild tomato species, which actually is a very interesting story, which shows us how acyl sugars have evolved in Peru and Chile across the Andean mountains over an ecological gradient. So if we know the fragmentation patterns, we can look at the compounds with more confidence in sort of a semi-targeted metabolomics. But what if you don't know the fragmentation patterns or the patterns are not consistent? We have been using two different approaches in this case. First is just looking at identification-free methods, which like PCA or information theory-based methods, etc. And second, instead of identifying the specific compounds, we look overall at compound classes instead obtained through untargeted metabolomics. One of the experiments that Liz, a grad student in the lab, did was look at Brachypodium distachya. This was a project funded through Joint Genome Institute. And we grew Brachypodium under several different conditions, not necessarily stress, and did RNA-seq and metabolomics from all of those samples. All the pre-processing was done, and then we analyzed the data in more detail. So I won't go into all of the data analysis. All of that is published in a plant journal paper, which can easily be found. But I'm going to talk about this information theory-based metrics that we use. So here we looked at two metrics, diversity and specialization. So diversity is a metric that's used. It was originally developed for transcriptomics back in 2007, and it comes from information theory literature. But essentially, if all of these are metabolite features with almost similar representation across all the samples, then the diversity is quite high. But if one of the in treatment two here, some of the metabolites are highly enriched, and their contribution to the overall signal is much higher, the diversity of the sample decreases and you get more specialization. So with that, we can look overall at the diversity and specialization of all the tissues and conditions that we looked at. One of the patterns that we see, which is also replicated in the negative mode, is that leaves are much more diverse in their metabolome uh, compared to uh, compared to roots, which are down here. We also see this big increase in specialization if when the roots are subjected to mycorrhizal symbiosis, and that's probably because of the mycorrhizal metabolites, the fungal metabolites that are also there in the metabolome sample. One interesting pattern we saw here was in this hydroponics experiment where we grew the plants under regular conditions as well as heat stress, as well as heat and no copper combined. The heat stress was much more resulted in much more specialization um, compared to uh, compared to just heat and no copper together. This is another metric called relative distance plasticity index, or RDPI, that that's kind of bypasses the differential expression analysis. So in differential expression, we have to apply thresholds, twofold upregulation or 0.05 pause discovery rate. RDPI takes into account all of the possible changes up and down together compared to the control. So you see no controls here on the axis because each test is compared to the control in a pairwise manner. So each dot here represents each pairwise comparison across all the replicates. Um, we see here that leaves do not change much under the stresses, under the conditions that we applied, we grew the plants in. Roots show a significant perturbation of their metabolome, and heat stress under the root had the maximum perturbation, and heat, no copper, in fact, no copper sort of primed the plants to tackle the heat stress. We see the same thing in the transcriptome too, that the no copper condition for subjecting the plants for 1 week without any copper in the medium primed the plants for the heat shock that we applied after after the stress conditions. And we do see that these have some phenotypic correlates. We replicated an experiment at the phenotypic level, and we do see there's a phenotypic impact, and we have some hypothesis in the paper about why this might be occurring. Now, going back to the question of predicting structures or identifying what is changing, we use Canopus, which all of you would know is a machine learning based method for predicting the classes of compounds. And we had the advantage of using a joint JGI machine with Trent Northern and Ben Bowen, who had run thousands of standards on the same instrument. So comparing the performance of Canopus with those standards and the classes of those standards, normally with Canopus we can get 80%, 85% of the peaks with the features with some class annotation. But if we take the FN score and only look at the classes with high FN scores, um, then that number actually reduces down to 40%, where we can say anything, where we can predict the class of the compound with confidence. So we proceeded with analyzing the 40% of the peaks that could be annotated with Canopus and looked at their overall RDPI, how much they are changing under given stresses. Now, this is a very complex figure, and that represents a challenge with analyzing class-related data. But I'm going to focus your attention on this two examples here, which shows what sort of information we can derive from such analysis. Flavonoids here in the roots were significantly perturbed compared to the leaves. And again, in furanoid lignans were important in this case in only under heat stress conditions, but not in other conditions. So they might have some functional roles there, and we also have transcriptomic data that we can dig into further and see if the metabolic pathways for these compounds were also changing under the same stress. So for RNA-seq, we saw flavanol biosynthesis significantly downregulated in the roots, and here we are seeing perturbation, which includes both up and down, and RNA-seq showed a downregulation in the pathway. Now, one of the constraints that I felt when we were doing this analysis was we only had the structural predictions, and based on that, we were inferring the function, which was not necessarily one-to-one. Those flavonoids have positive effects as well as negative effects. Some of them were going up, some of them were going down, and that's partly a problem with the fact that is due to the fact that we are only inferring the structure of the compound. What we are doing now is using large language models to look at several different features of metabolites. So we are developing this plant chemical ontology, as I'm calling it. We're reviewing thousands of papers published since 1980s and assigning metabolites to various different properties that we can derive from papers. Eventually, we will have something, a knowledge graph. We are doing this project in collaboration with David Wishard's lab at Alberta. And eventually, for example, if you find a metabolite that's upregulated in your data set, you may be able to compare it to our database and see if that is known in other species in the same tissue expressed under the same condition and so on. Yeah, so that's about LCMS data, and I ended this part talking about how we are using large language models. We have quite a bit of experience with working with large language models in this next story that I'm going to talk about on predicting enzyme function. So just like metabolites, that fact that less than 10% of the metabolites we can annotate from LCMS data, the same problem is with enzymes, which generate this metabolic diversity. Although they generate the diversity, the functions of most enzyme families in plant genomes are poorly annotated, and that's true for many, even in mammalian genomes too. What we reasoned was, if there's an enzyme family where you want to predict the function of a tomato gene, but only the rice and the Arabidopsis enzyme functions are known in this family, you cannot predict the tomato function with confidence. It's because in this intervening evolutionary time, there would have been duplications, gene loss, promiscuity exists in enzyme families, and that would result in either missing annotations, incomplete annotations, or wrong annotations. So the hypothesis here was if we increase the functions of the enzyme family members across this functional resolution, then we may be able to predict the tomato ortholog with greater confidence. For this, we are using the BHD acyltransferase family in plants as our model system. So this family transfers an acyl group from a CoA donor to a hydroxyl group on an acceptor. And the acceptors can be very varied structures. They can be aromatic, aliphatic, amine-containing, and more complex structures as shown here. There is also the diversity in the donors, the two substrate reactions. So the donors are also quite diverse here. These compounds play an important role in various different plant processes, from growth to development to stress response, reproduction, etc. In plants, because of all of these diverse functions, the number of enzymes per genome has grown from 1 to 2 enzymes in algal genomes down here to dozens and hundreds of enzymes across plant genomes. In tomato, there are 100 BHD enzymes in the genome, but all except about 10 of them have this kind of an annotation, HXX3 type acyltransferase, in the current genome version, or transferase and acyltransferase. So the substrate class of this enzyme is not annotated. We took a decision early on to only focus on substrate class because, one, it could probably be connected with metabolomics more efficiently, where class prediction is better. And second, enzymes are promiscuous, so they may use compounds from the same substrate class very easily compared to, that is a simpler problem to solve than predicting the specific substrate of the compound. Now, if we go to the UniProt database, which is the store of all annotated enzyme function, protein function, only 62 BHDs across all plants have been deposited in UniProt. That creates problems with several downstream uh, biological processes. And this is partly because there are only 2 plant curators at UniProt. There are 20 for biomedical, mostly human literature curation, but only 2 plant curators, and they do their best, but they can't keep with the pace of publications that exists now. So we thought we would help them a bit. In 2022, we published this paper where we are looking at Lars Cruz, a postdoc in the lab. At the time, he manually compiled papers and enzymes from several different species and put them in a phylogenetic tree. Manually, we defined 11 different classes of these substrates and ended up predicting their function based on orthology. But soon we realized that if you want to scale this, the problem is quite immense and we can't do this manually. So we published this paper called, developed this tool called FuncFetch, which uses GPT-4. And it goes through several steps. Some of it is manual, some of it is computational, Python-based, some of it is these orange-colored steps are the ones where we use GPT for reading papers and for extracting enzyme substrate interactions from those papers. We were able to apply this to 9 different enzyme families, and at a cost of $65, we screened 27,000 abstracts, and from those we selected about 5,000, 5,500 papers, and from that we isolated about 32,000 enzyme substrate interactions across all these 9 families. In BHDs, we were able to take it to do manual curation of the extracted enzyme activities. We assigned SMILES, PubChem IDs, etc. And we have submitted it to 3 different databases where it can then be applied to other other data. It can be shared to other databases or people can use it more readily. This is obvious, but compared to 3 human readers, the time required for FuncFetch is an order of magnitude lower, and it can be scaled up without getting tired. Compared to what exists in SwissProt, we were able to get 3 to 4 times more papers, more species, more acceptors, donors. Everything was more compared to even our previously published paper. Yeah. This then we organized into orthologous groups. This is a gene tree of all the characterized BHDs in our data set and the substrate classes that we obtained from NP classifier mapped onto these classes. But main thing here is if we put it into orthologous groups, then we can query these more efficiently. So previously, I mean, normally what people do is to find out enzymes involved in their favorite pathway, they would look at differential expression, for example, or QTL mapping. They would get hundreds of genes. Then you identify the enzymes of interest based on your chemical structures, etc. Now you might find 20 BHDs. Those 20 BHDs you would literature search to find what is previously known about those characterized enzymes. You would download the sequences, make phylogenetic trees, and then you would select the enzymes for experimental validation. But now, with at least for BHDs and hopefully for the other enzymes soon, we can, researchers could just upload their FASTA file onto the website and can know if any of those have previously characterized enzyme activities. So this is an example of our website. You can go to tools.moghelab.org, and this has this tool where you can upload your sequences, and if you blast it, it blasts against the orthologous group and can tell you, okay, this particular enzyme is conserved across all of the land plants, many different species. And the closest hit is with this HQT enzyme that is in these two papers here, they used caffeine-CoA, quinate, shikimic acid as substrates, acceptors, and donors. So the researchers can go back and design their enzyme assays more efficiently. This is another enzyme that is conserved only in dicarts, and it's involved in anthocyanin acylation. This was an unknown enzyme that we knew that is present in tomato but is not annotated. Uh, this tool revealed that it's only present in Solanaceae. The closest hit was this with Solanum richardii with 70% identity, and it told us that this enzyme uses spermine and spermidine as substrates with caffeine-CoA, coumaryl-CoA, ferroyl-CoA as donors. So we cloned those enzymes from tomato, we did enzyme assays, and we do see expected activities compared to the empty vector controls. Okay, so in the tomato genome database, we have 10 BHDs with annotations. In our previous paper, we could increase that annotation to 42 BHD. So this is only the substrate class of the enzyme. And with this phylogenetic approach, we can increase that number to 54. So this is not just in tomato, in also soybean and rice, we were able to increase the characterized enzymes to more than 50%. And we are currently applying this to 87 high quality plant genomes across the plant kingdom. The next question is, why stop at 50%? Can we increase the proportion to 100% of PhDs? And this is where the new tools in AI come in handy. So we are using evolutionary scale model to go to the remaining 100%. And just to give a quick example of how ESM works, this is using large language models. Large language models, if the model is trained properly, then if you mask a given word, for example, if there's a missing word, if it understands the context of the entire paragraph, the sentiment behind the paragraph, then it should be able to fill in the missing word very, very accurately. And the same thing is for next word prediction. If it understands what the sentence is, then it should predict the next word very efficiently and accurately. ESM3 also does the same thing. ESM3 takes structure, sequence, and function tracks as inputs, and based on its embedding structure, it's able to complete the missing sequence, structure, function in its own domains. The problem with ESM3 is it takes the embeddings and then predicts interpro annotation with those embeddings, which we already know from protein PFAM annotations that these are, you'd know what sort of annotation, or it's a BHD or P450 and so on. So this is not the output. The standard output is not really useful to us. So we took those embeddings and developed our own classifier to predict whether the enzyme uses aliphatic donor or aromatic donor. And as you saw in previous examples, the enzymes can, if they use caffeine-CoA, they might very well use ferroyl-CoA or cinnamoyl-CoA too as donors. And so, the same thing is with aliphatic. So in this case, we just focused on these aliphatic donors, aromatic donors. And the performance in all of these cases is pretty good. AUROC, precision recall are pretty good for this kind of classification. If we just focus on the donors, we can get almost all of the BHDs fully annotated with aromatic or aliphatic donors in all different species. So that's a good thing. But when we try to use acceptors, previously, one question is how do we develop a classifier for acceptors? We need some classes. Previously, we did this manually by some binary classification rules, but now we are using NP classifier for doing this. We found that this is a little better at natural products compared to ChemOnt. And so we are using NP classifier for our classes. Sometimes, in most cases, classes, sometimes we use the pathway or the super pathway annotation. Now, the performance for donors is pretty high, but performance for acceptors here is not that great. And this is partly because NP classifier classification itself could be a problem. We use leave one out uh, uh, testing for getting the precision recall. And in this case, if enzyme is predicted to use a flavonoid, but if it's predicted to use isoflavonoid, um, then it would result in a negative uh, classification. So uh, in this case, just because the structures are so similar, the algorithm can get confused. And it gets confused very often between structurally similar compounds. So here you can see this is basically plotting the activation of the ESM predictions. And if the color is yellow, that means that that's where it's most, I mean, that's the next highest probability. There's a top class and the next highest class um, that the activation would be. So it frequently gets confused between diterpenoids, taxane diterpenoids, tetracycline diterpenoids. It frequently gets confused between flavones, isoflavones, anthocyanins, flavonoids, all very structurally similar compounds, or shikimic acids and simple phenolic acids. These are very similar. So these structural classes, um, I mean, I personally don't consider this to be a wrong prediction. In this case, what we will do is just provide all of the predictions to the user, and they can make their own decision in this case. We are currently doing this. The next target is UDP glycosyltransferase, which we have finished curating completely, and in the process of going through this process. And this is all plant UGTs specifically. The rest of the enzyme families, we are curating them through computational means currently. Okay, so to summarize my talk in one slide, essentially, if we are talking about enzyme function prediction, I think the problem will be solved soon. It might already have been solved soon. There are tools like Enzyme Cage, which was recently published. There is Easy Specificity that is published. We haven't used those tools. Some of those are behind paywalls. AlphaFold 3 also has their own proprietary system, which we have. It's behind. It's proprietary, so we can't even use it. But those that enzyme function problem could be solved very soon. It might be, for linear specific enzymes. I'm not sure how well the performance would be. That's something to be looked at. And if the enzyme prediction problem is solved, then pathway discovery, pathway engineering could become significantly easier. The problem of metabolite annotation, in my opinion, is still an important challenge to solve, um, and better in silico spectral databases, as well as better and more annotated MSMS spectra in different modes could be very helpful in addressing this challenge. That's it, and I can take any questions.
Yeah, thank you so much, Professor Moghe. This was very illustrative, very informative, a great presentation in terms of the different approaches you've shown to us. Yay.
Thank you.
So, so I invite everyone to just unmute yourself, or maybe you want to show your hands and then ask questions as you feel appropriate. Is there anyone who wants to take a first step?
Oh, yeah. Hi, very nice talk. It's very interesting. I mean, the using AI to do yeah, such a great job. And my question is, now we know AlphaFold is very powerful, and do you think it's useful to use AlphaFold to get the enzyme structure and then implement that structure information to the function prediction?
That's exactly what we've done using AlphaFold structures to train our to obtain these predictions for the enzymes for acceptors and donors. So AlphaFold, there were some issues with the previous version of AlphaFold, that the resolution was not very high. It wasn't performing so well with lineage-specific enzyme families. over the years, I think with AlphaFold 3, that the resolution problem was definitely solved, and then the structural predictions with tended to be a little better. Yeah, I think it is still a valid approach. If you are going to predict specific molecular interactions, like specific amino acids that are responsible for the enzyme substrate interactions, that's something that would need to be tested in more detail.
Okay, and also if the substrate, the absolute chemical structure of the substrate is not fully determined, I mean, maybe there are some stereoisomers for the substrates. And do you think AlphaFold can still predict the function very accurately? Because from MS2, I mean, many structures cannot be determined, absolutely determined.
Yeah, so there are different approaches that people have used in easy specificity or enzyme cage, where they actually predict the substrates. They see if the activity is there in the top 5 or top 10 of the predictions. That if it's present in the top 10 of the predictions in enzyme cage, they consider it a success. And yeah, so it may not be the top prediction, but maybe in the top 10.
Okay. Yeah, thank you.
Further questions by people? Anyone who wants to take the next one? All right. Well, I have lots of questions, so I can talk forever. So the first one, you focused on function as just a biochemical function, right? But biochemistry, is fun, and we want to know who's doing what. But what about physiological or biological functions? So can we predict from structures what the purpose is? Like, is it defense, communication? Is it, signaling? Can we say anything about these types of enzymes, the compounds they produce, so that we also might improve our understanding of biological functions and not just biosynthesis?
Yeah, absolutely. So it's going to be a very difficult challenge, and I don't know if it is if we are capable yet to solve that, because compounds have very context dependent functions, and the function in the root will be different from that in the leaf, tissue specific, cell type specific. So as a first step, we want to build this knowledge graph of all the functions of metabolites, and that might tell us that could be a starting point for. I'm not sure if anything of this sort has been done in humans, but there is also so the idea is, if we have this database of plant metabolite function, we may be able to start predicting a function of the metabolites with more confidence. Again, that would still be biased on what is known. Yeah.
Okay. All right. Well, then, going back, I understand it's difficult because, as you say, it's context dependent, and, you have focused here quite a bit of abiotic stress, but other people, including other people at Cornell, they say, the secondary or the specific metabolism gets specifically active under biotic stresses. So when caterpillars bite and when, you name it, right? Have you tried to elicit bigger responses that way to get a more idea of the metabolomes under these different biotic stress conditions?
Yeah, no, we have not worked with biotic stresses as much. Yeah, but this diversity specialization is a metric that has been discussed in this context. Essentially, whether plants mount a specific response to stress, or whether they constrain their metabolism to produce one defense metabolite in large amounts, or whether they just mount a generalized response and try to kill the pathogen or the pest through everything. And yeah, Ian Baldwin's work in chemical ecology or Emmanuel Gacurel has done a lot of work on this topic.
Hooshu, you wanted to have another question? Follow up?
Oh, yeah. Because I'm thinking about this method as the prediction of enzyme function sounds like a very general approach, because it's also important to predict the enzyme, for example, in human and animal. So that's so then my question is there a difference between predicting the enzyme function in plant versus predicting the enzyme the enzyme function in animal? Or is there any special procedure in your method that has been tailored for the plant enzymes?
No, I don't think there is anything different. Plants tend to have lower amount of data than humans, generally speaking. The family that we work with, both BHDs, UGTs, some aminotransferases, are very plant specific. So we don't have enough structures in those. And so the AlphaFold also structures tend to be less accurate for those enzyme families. You might in fact have more accurate structures in humans.
Okay. Oh, okay. Thank you.
So then going back to biochemistry, and as I said, if nobody else, has questions here, I will, have more. So the biochemistry and the functions of those is of course also context depending, like cell dependent, right? So if an enzyme doesn't even see a substrate, if there's no transporter, then you would never know, right? It might not even do the production of a predicted substrate or reaction pair, or you say acceptor donor pair, right? So how do we tie in the transporters here or the cell specificity?
Yeah, I had asked a graduate student to look at tissue specific expression of genes, but she got distracted doing some other work later. But the the grand idea was to be able to use these DNA foundation models, for example, to predict the expression of genes. And yeah, it's I think there is a lot more work that needs to be done. People can predict the expression level of genes more accurately. Whether a gene is high or low, that kind of classification can be predicted. But the tissue specific, cell type specific expression is difficult to predict, at least in plants right now. But yeah, combining spatial data and enzyme data, we might in a few years, I think, have a way of integratively predicting enzyme function.
All right. Are there people who would like to raise their hand? You want to have a question, please feel free to do so. It's now the chance to talk to the speaker, because we don't, this is a virtual meeting, so you have to just, okay, Shen?
Yeah, I'm thinking about the verification part. I think it does have a verification steps, and I would like to know more. How do you have like ground truth or true positive or true negative? Or is it by biosynthesis, multiplexing, or even when you are doing the ChatGPT AI for large, like 27,000 papers, how do you use such QC to guide the training is working to give you a reliable result?
So are you talking about the enzyme part or the metabolite, this metabolomics?
Kind of both. It kind of goes to, one, there is like the steps of at the end, it's verification. So I wonder what kind of verification. Okay. And the second would be the ChatGPT data mining. How do you have a knowledge check saying it's doing the right thing? Yes.
So in this case, this was the early years of ChatGPT-4. We actually have a figure in this paper showing how in the 6 months when we were doing this project, ChatGPT went through 3 different versions, and we could see in real time the accuracy and the precision and the recall increasing in all cases. Because we have the ground truth from our previous study, we could always compare it to this study, which was already published, and see if the extraction is accurate or not. If it's extracting the correct interactions. What we see is it's able to extract the substrates pretty well with high accuracy, 0.8 or so precision and recall, but it cannot put the acceptors and donors together. Because that might be sometimes in a figure or they might be in very different parts of the manuscript. It gets confused to put those pairs of substrates. With that, the precision was only 0.4. We verified this, a lot of this was manual and using our in-house Python scripts.
Can I also ask, like, when you present metabolomics data versus transcriptome data, like, what would be approaches beyond calculating correlations?
That's a good question. Yeah, we have there's co-expression which is still based on correlation. Overall patterns as I showed, but I do not have a good answer to that. It is very project specific, context specific, I would say. Pathway mappings using KEGG or other approaches.
Or even with the correlation, have any verification validated saying, oh, it is the transcriptome and this metabolites that's mapped together, and we see both upregulated in the root, for example.
Yeah.
I'm also here reading out a question that has received us via chat from Kira Tietke.
Mm-hmm.
Thanks, Gaurav. She is traveling right now, so she missed how RDPI is calculated. And the reason why she asks is, if roots have fewer compounds than leaves, isn't it then expected that relatively more changes are found in the roots? That's Kira's question.
For the second part of the question, Kira, I'll have to go back to the data and see. But RDPI is basically if you have 5 replicates of the control and 5 replicates of the test for each compound in the control and each compound in the test, you are basically calculating the difference for that same feature in every pair and then averaging it across that pair. And that's basically being plotted across all the different pairwise combinations. If there are 5 and 5, you get 25 points that you're plotting for each, the average RDPI for each pair of sample.
Mm-hmm. Okay.
Yeah. For the second, I'm not sure. Yeah, I'll have to think about it.
Okay. Well, it's just by the combinatorial calculations, right? You have fewer compounds, so there are fewer chances to have correlations in a way, right? Anyway, so I also wonder about mass spectrometry a lot, right? And you said, well, it would be nice to have better predictions of mass spectra. You said in silico. Well, this idea of in silico predictions of mass spectra is around for many decades. In fact, people have published tools that just don't work. And they're just giving far too many false positives. What are your thoughts? Why is that the case? And could AI improve that conundrum?
Yeah, that's also a good question. One of the first projects we started was trying to predict lipid classes using feature-based machine learning, random forest, and so on. It works beautifully for lipid maps, lipid blast, 30 classes, 8 classes in each of those sets. When we applied to actual mass spec data, it failed dramatically. So I think why it does not work, it's It's not, partly it could be noise, it could be instrument variation. There is positive negative mode variation. It could be that the fragmentation characteristics of specialized metabolites are not well understood in complex matrices. Yeah.
Okay. I have a very simple question. Why do we have 100 of these BAH acyltransferases? I mean, why are there so many? So we also had, in another project, we looked at UDP glucosyl and there are also over 100 in one species. Why do we have so many?
Yeah, why is the most difficult question to answer in this case. Yeah, here they, partly that they are important for various processes, and so some of them tend to get specialized, and there is no fitness consequence to their expansion. So I think that there are two ways to look at it. One is the adaptive explanation, where this large copy number is important for some reason. Second is a drift hypothesis, where just because the genetic space is large, they tend to get duplicated more frequently. And unless there is a cost that exists for not having those, they would keep increasing. So yeah, these duplicate genes tend to get duplicated more often, and they tend to also get lost as often. But they play important roles in several processes. They tend to get plugged in different processes too.
So you've looked at tomato. Yeah, and I like tomato. I like potato. Can we predict the metabolomes of foods? You get what I'm saying. So we have this periodic table of food initiative where people use mass spec just to enumerate what are the compounds we find in foods, right? That's the part from the mass spec side. But, how approachable is it to try even to do that for a diversity of foods?
For even one food, this is an example. In tomato, we using MSMS networking with GNPS, we can get less than 10% of the peaks. Some of those would be adducts and in source fragmentation and so on. And with all of that, with MS2, the tool that Ming has developed recently, where you can predict the moieties that can be expanded on compounds, it still goes to 15% maximum. So if 85% of the peaks or features cannot be annotated, I'm not sure how we can predict the entire metabolome.
All right. Again, asking, there's Kira has a follow-up question. So she asked, about your first project. Do you expect overall diversity of the RGs to be important for nematode resistance, or rather some specific RGs?
That's a good question, Kira. We wrote a current opinion paper on this, on whether the diversity is important. It's the same thing as the enzyme family question. Is the diversity important? And there is some evidence that it is important. Like the specific acyl chains, you get more damage on fungal cultures if you use one resin glycoside versus another resin glycoside. The same thing with acyl sugars. Some diversity is important. Ian Baldwin has done beautiful chemical ecology work on this topic. But that might be even if we have 300 different structures, 90% of the signal is by 5 or so resin glycosides. The rest of them are just, they exist and we can detect them because we are using LCMS, high resolution mass specs. So those others could be the promiscuous activities that just exist. And the same question can be asked for transcriptome. If you do Illumina, you see many, many more transcripts than genes. Now, are all those transcripts important? Likely not. This is probably, you don't need to perfect the system if there is no functional cost to it.
Mm-hmm. Okay. Sina, you have opened your screen. Do you have a question? We do not hear you. You are still muted.
Sorry, I just completely forgot about this session. I just came in now.
Oh,
I
I'm
know
sorry.
the start.
Okay. Okay.
Yeah,
We are just finishing up with questions.
yeah.
Yes, any other?
Sorry.
And this will be or was recorded. So Gaurav, do you have your permission to put it on YouTube?
Yes, that is okay.
Okay, so Sina, it will be on YouTube, so you will be able to watch it later. It'll take usually a week or 2 weeks until it's up on YouTube.
Thank you. Sorry.
No, it's okay. We're all in the same boat here. Are there other people who have burning questions right now? That is not the case. Well, then I would like to thank our speaker so much for your contributions, for your openness to share, for your research. Congratulations. So thanks everyone for joining us today. And we will make sure that you get the invite to our next lectures as well. Thanks everyone for coming, and hope to see you soon.
Yeah. Thank you, Oliver, once again, and thanks for this wonderful session and all the interesting questions.
Bye-bye.
Thank you all for coming.